So, welcome to my dear student. Basically, my this video is for my two research scholar from North Macedonia who are working with me for cancer therapy, especially cancer immunotherapy. So, let us start the class. Actually, nowadays I am so busy with my uh, prime responsibility that I am in round the clock shift. So maybe I'll for next 6 months I may not be regular, but definitely the foreign students can ping me in WhatsApp and with your uh, queries, and definitely I will try to sit with you uh, based on my convenience. So today we are going to discuss a technique which can boost the immunotherapy. Okay, which is called hyperthermia. As the name suggests, that basically it is to elevate the temperature. Okay, just to increase the temperature. So, uh, this hyperthermia is a medical treatment that involves raising the temperature of body tissue to help uh, destroy the cancer cells. The temperature can be elevated up to 45 degrees Celsius, and this will excite and cause the death of the poorly developed um, uh, proliferating cells. Okay, so this is basically the hyperthermia. Uh, so mainly why I am giving the glimpse of this, because we will do the simulation. Because uh, to go ahead with this kind of treatment, first you need to simulate that which, because the say somewhere the tumor is developed and nearby some very sensitive tissues are there. So once you are doing this hyperthermia in that, then you have to predict that whether this elevated temperature can create any harm to the surrounding um, uh, useful and sensitive tissues. So for that you need to do the simulation. Okay, so for that purpose only this video, just to give an idea how this hyperthermia is hyperthermia works. So for that only I am giving you very short. It's a very complex um, um, physiological phenomena that and physiochemical phenomena, so that I am not detailing that already. For the first year student, I used to teach this immune system. There, you can refer my classes there. Okay, so this hyperthermia or this temperature can be elevated using several technique, using radio frequency, microwave targeting that particular tumor, so that we will understand, or that we will learn in some other class using some simulation finite element modeling softwares. So let us okay, okay. Okay. So now let us understand how our immune system works. So when any allergen, pathogen or antigen, means which can create the disease in our body, when it is crosses our body barrier, uh, so our immune system and the complement system, it triggers, and using some signaling, it will accumulate all the defensive mechanism and the fighting element and instruct the immune system that that particular protein, or virus, bacteria or pathogen or antigen, it is harmful to our body, and kills. Okay. So these are the things, it is done by some chemical signaling and some pathway. Now the question is, how body, our immune system identifies our necessary tissues, which are like our all the muscles, say our liver, our kidney, that also having some protein. But how body recognize? So there is a pathway, it's called, uh, or there is a, you can tell there is a chemical process, which is called PD1 PDL1 pathway, and using this, our body can identify which is foreign and which is our own. Okay. So basically it starts in the lymphatic system. Our lymph nodes produce that tumor infiltrating leukocytes, or simply it is in T cell. Okay. So this T cell is the scanner for our body, uh, and there is a process in that T cell, in the periphery of the T cell, there will be a genetically coded receptor which is called programmed cell death protein 1, or PD1. This PD1 is, while this signaling mechanism take place, this PD1 always try to search its counterpart called PDL1, which is already available in the healthy cell. And once it is like, when you are crossing our international border, the security personnel, they will check the passport. So this passport is that PDL1. So once, and this PD1, you can think that this PD1 is the security system, so that will always try to see its counterpart PDL1 in the cell, in the cell in your body. Okay. So once it this PD1 able to see that PDL1, then it will recognize that this is not harmful at, and it ignores. Okay. And once this PDL1 is not available, normally this is not available in the abnormal cells or in the cancer cell protein, and it will destroy. So this mechanism, this I have a very good, I have done a very good work on this, and you can find that paper. It's a open journal. You can go to the PubMed. It's a National Library of Medicine, and the subject, and the title, the paper title is an extension of cancer immunotherapy with dostarlimab. It's a monoclonal antibody which can block, which can inhibit the PD1 PDL1 pathway for that particular cancer cell, and it is extensively used. Normally this monoclonal antibody, immunotherapy, it's called the immunotherapy. Now the question, why does our immune system allow cancer to exist? How it's getting proliferated? Because this cancer cell having a very special masking mechanism, that uh, they can they can produce a false PDL1. This is not exact PDL1. It's a false PDL1. Okay. So once, as I told, once PD1 will see the anchor protein PDL1 in other cell, it will allow, and they will think that it's our own system, own tissues. So this cancer cell having a false PDL1, and with that they can escape from our immune system and they can proliferate. Okay. So now the thing is, how to we have to find out some, or we have to augment, we have to enhance the security scanning of the T cell, or you have to do something so that this PD1 or the T cell can identify that false PDL1. How to do that? We need a trace of that. We have to find out that some special, like some terrorists are in the common gathering. So the army or the national security forces, they have to understand, or they have to find some methodology or some characteristics of the terrorist, so that they can easily identify and catch them and destroy them and kill them. Okay. So like that, we have to augment the scanning ability of the T cell, so that they can identify very easily the false PDL1 which is available in the cancer cell. Then our immune system itself can destroy the cancer cell, and it will stop the tumor growth and the proliferation. This can be, this ability, scanning ability of T cell, can be enhanced by applying heat to the tumor. Okay. So in this particular hyperthermia process, as already told, the temperature is elevated up to 45 degrees centigrade, which is considered as our fever. Will stress the cellular function of an already poorly developed cell structure, causing the death of that particular cell. Okay, simply you are elevating the temperature of that particular cell, the tumor area. Okay, it's a guided and focused heating process to that particular tumor area. Okay. The healthy cells, they can quickly recover from this stress due to the heat, but when this, due to that elevated temperature, when this cancer cell cause death, so it releases one special protein which is called heat shock proteins. This I am telling very common language, so that other non-medical people also can understand, but this involve a very complicated chemical process that I am not detailing, that already you people who are working with immunotherapy, or who knows immune system in details, they can easily understand. Okay. uh, so what I was telling, during this cell death of that particular tumor area, it release some heat shock protein and some antigens, and that molecules basically containing the specific information about the cancer cell. This heat shock protein and the antigens which are released during the cell death of the cancer cell death during elevated temperature, that having a special identity, that having some property or some information, that by seeing that our immune system can recognize that it is from the uh, cancer cell. Okay, and this, if our immune system can accumulate this stress and can modulate the T cell accordingly, or train the T cell accordingly, that you can, with this you can, with this particular information you can see, even though cancer cell providing that PDL1, but still, if the T cell can be trained with the information which is, which could be accumulated from the heat shock protein and the antigens released during the cancer cell death, then now our T cell is more powerful, and they can help the immune system to unmask the cancer cell. Understand? So why hyperthermia is required? To boost the cancer therapy, immunotherapy, that what is called chemotherapy. Okay, so basically, by elevating the temperature, we are causing the death of the particular cancer cell, and during the cancer cell death, it is releasing some heat shock protein and the antigen, which having some very specific information. With that, our immune system can recognize the cancer cell, even though it produces the false PDL1. Okay. So now what is, once the cell death, the cancer cell death occurs, and it releases that heat shock protein and the particular antigen, or the dendritic cells accumulate these proteins and return them to the nearest healthy lymph node. Okay. Now the heat shock protein modulate the tumor infiltrating leukocyte production to a newly evolved T cell that can now detect the hidden cancer cell. So job finish. So we are boosting, basically the hyperthermia is boosting the scanning capability of the T cell. Understand? So this is hyperthermia. This is the role of hyperthermia in cancer treatment. It is not only it is work for boosting the activity of T cell, but it is also helpful um, uh, uh, to prevent the metastasis. That means the cancer tissue can, mainly in case of breast cancer, metastasis is a very serious concern. So that tissue, because those are very light and soft tissue that can be come out from the cancer site, and it can travel with the bloodstream to the other part of the body. Okay. So this heating process increase the blood flow to the tumor, allowing more T cell to go there and fight against the cancer. Okay, and it is also it is found, and it is an additional aids to the surgery by treating the cancer, as well as in the chemotherapy, radiotherapy, which is basically done by sensitizing the tumor and boosting the effective dose. Uh, so this was, I'll show you one case study. So this is a picture of rectal adenocarcinoma, where you can see in the first picture there is a tumor, and the dostarlimab, or this monoclonal antibody, which is helpful for inhibit the PD1 PDL1 pathway for the malignant cells, and with that hyperthermia is applied. And you see, this is the picture after 3 months, and this is the picture after 6 months. You can see the entire tumor is vanished. This is for rectal adenocarcinoma. So in the next lecture we will be or the next class we will learn um, how to simulate this hyperthermia for a particular location of human tissue. We will be using some finite element modeling tool, and the particular module, like in case of very preliminary heating experiment, we can use the heat transfer in solid with the specific property of that particular tissues, and then we will learn using radio frequency heating or microwave heating using ACDC module in some finite element modeling software that we'll learn slowly. So this is my lunch time, even though it's late. It's uh, crossed 3 p.m. in India. So I'll be going for my lunch. So hope the video will be useful to you. Always you can ask me if you have any question. Um, if I am not available, you can always you can call my WhatsApp number. I'll try to attend. Um if I am not available or I am not reachable, you can drop your queries in my WhatsApp, and I will answer you accordingly. Thank you so much. Thanks for your patience.